with, again, introduction. As I said the other day, the initial 10 lectures basically are introductory immunology, and you will see quite a lot of overlap from physiology, pathophysiology. But my advice to you will be that you need to read the book, and the way the PowerPoints are written, are not complete. They're totally incomplete. They will just encourage you to go back either before or after the lecture to prepare yourself for the concept. Okay, and as I said that while I'm talking, I am recording the lectures, and they are going to be posted. And as a matter of fact, if any of you is interested and does have an ability or knowledge of all these kind of recordings and so on and so forth, you're most welcome to come and see me after the lecture. Because it takes a while, and I did post it on Moodle, but I think the files are too big to be downloaded from Moodle. So we came up with this YouTube channel, private channel, but still there are some glitches, so we are still working it out. So if you do find any need for help, you can come and talk to me. And as I said, that if there's any burning question, you can write it on a piece of paper, pass it to me. Otherwise, leave the questions to the end. And thirdly, I have not changed the lectures from last year. As I said, the book is available online. It's a free book. You must have seen that. And I'm also reading the new book, but since it's a basic introductory immunology, I don't think that it's going to be much change, but definitely in the second half for the clinical immunology, you would see the difference. So we have plenty of time. As you are reading, I'm reading as well at the same time. So the basic family of immune response, if I was to put it there are two players. It's an antigen and antibody. So that's the basic thing you have to understand. And everything goes from there, right? And as we go along, we may have some workshops, so you can try. It's a boring subject, it's a dry subject, so you can make up your own mnemonics, and you can make up your own kind of interest. And if I was to say it's a romantic love story, and antigen will be the boy, and antibody will be a girl. And you will see why do I say that as we move along, just for the concept point of view. Okay, that's where the family begins. So you can come up with a lot of mnemonics. I do have some of mnemonics from the last class to make a hard concept go into your mind, and you want to keep it for good, because all the tests are based upon this concept: antigen, antibody. And we have a full lab. One of the chapters for immunological technique would not be covered in the lecture. We will discuss that in the lab, but I would encourage you to start reading on that, an antigen antibody interaction. That's what the story is. Today, continuing from last time, as I said, innate and acquired immunity. And I also told you your book wants to call acquired immunity as adaptive immunity, pretty much the same thing. We talked about some of the cells involved. We'll go in a little bit detail. And today we'll begin with a story of an antigen. So what is an antigen and how does it get into the system, so on and so forth. So that's where we left. I told you that innate immunity is that you're born with this immunity. It's a hardwired thing. And basically, it's non-specific. It's going to fight off everything. It doesn't really care. Because if you've been vaccinated with MMR, measles, mumps, and rubella, and if any of these three microorganisms want to get into your system, these innate immunity barriers will be there, but not sufficient to fight off the disease. That's where we need acquired or adaptive immunity. So that's what something you have to keep in mind. So when I say innate immunity, these are physical and chemical barriers. So your skin, your mucous membrane, there is a fatty acid on your skin. So if you wash yourself too much, you're hypochondriac, so you're gonna dry your skin up and you expose your skin and prevent the skin from having that barrier. Okay, if you have a cut on the skin, burn on the skin, you open up the front for microorganisms to get into the system. Okay, but at the same time, your clothes, your jewelry, your cosmetics, your contact lenses, anything that you want to bring close to your skin will be sampled by the immune system. Immune system is looking at it. That's what I'm going to talk about in detail. It may decide to react and you will have allergies. It may not decide to react. It's not going to react to gold, for example. Gold doesn't have good, so any allergic reactions. Other than that, and some other heavy metals, other than that, everything that you wear in different parts of your body, on the body, touch, whatever, is going to provoke and incite a reaction. The very first defense that you have is your physical barrier. The saliva in your mouth. There's a pH in your mouth. There's a pH in your nose. There's urethral pH. There's vaginal pH. There's rectal pH. So these are some of the chemical barriers that we do have to protect us from impending danger of microorganisms which surround us at all times. There's nothing you can do about that, but you have to be vigilant, careful, but as I said, that you don't want to be a paranoid person. Now, the other concept we did discuss that was intra and extracellular killing. Doesn't mean that body is going to wait for that autoimmune response. The killing by our cells, defense system, can start non-specifically at all times. If you remember, when I taught you pathophysiology, I told you necrosis and apoptosis. Okay, so these are the important things. Now, cells are an important part of immune system. There are tons of cells, and no wonder they are there for a good reason. They have a good name, and many a time the name speaks for their function. And as we discussed yesterday, when I say cellular immunology, I said two important branches: humoral or humoral, that's an antibody mediated, and cellular, this is cell mediated. And again, you can see from here, right from the stem cell on the top, there are lymphoid cells that go in a series of lymphoid cells. There are myeloid cells, there's a series of myeloid cells, and you can see all of them basically have a function. Something on the right that you see myeloid progenitors, over there are the cells that we talked about in neutrophils, granulocytes, red blood cells, and plate cells. They do play a role, but this role basically, for most part, is a part of innate immune system. So if neutrophil is going to go and kill a bacteria, it's not going to ask bacteria to give ID. It's not going to look for any ID. It's going to be non-specific random killing. Whereas if you go towards the right-hand side over here, these are very specific lymphoid cells. These are T cells and B cells, and you can see they are the parent cells. And as we go along further down, they become very, very specific, and you must have heard of CD4, CD8, and tons of other cells. I also talked about antigen presenting cell, as the name suggests, antigen, so I'm going to discuss that today. And again, you can see some of the cells are macrophages, dendritic cells, mast cells, and many more cells. So keep in mind the names of all the players, right? So these are important. And then again, you can also see that these are made in bone marrow, and these are in the periphery. It could be in the blood, it could be in the tissues, and so on and so forth. So of course, since this is a basic immunology class, but keep in mind things can go out of control, and each and every cell that divides, if I tell you all these cells divide and proliferate, so what does it suggest to you? Hmm? Cancer, exactly. So all of them can become cancers when it goes out of control. Okay? So I would want you to memorize the shape of the cells, because as we go for reactions, remember one thing I said yesterday or day before yesterday, uh, that most of the immune cells want to have a physical contact most of the time. So they really want. So if I say the antigen antibody are the major players, so they basically want to come in contact with each other. So they have to physically bind to each other. We'll discuss what kind of bonding that's happening between these two cells. Okay, but these are the developmental pathways of the various types of the cell. They all start from the stem cell. You pretty much know. But also keep in mind that this is just for our learning, that we divide them into different uh, kind of groups, but in terms of immune response, innate or acquired, but there can be an overlapping zone. There can be, the cells can help each other. So keep that in mind. It's not very, very restrictive. The other important thing that you can see from here, that it says over here that these are restricted in the blood. So neutrophils, uh, monocytes, platelets, and all these cells you mostly should see in the blood. But if you were to see neutrophils in the tissues, that's not normal. Okay? Because if you may see that. So this means something is going on. It's just like uh, a crime scene. So you see all these barricades over here. Something unusual is happening. All of a sudden you see the tape, and then something's happening. So that's something is going on. So keep that in mind. Normally they should not. Remember when we talked about migration in pathophysiology, I told you the steps when the neutrophils marginate diabetes and go into when they're called upon duty, when there's infection there, there's a repair there, there's something happening. Something has to happen, because these cells carry tons of weaponry in terms of reactive oxygen species, reactive nitrogenous species, which basically are destructive. Okay. Now, I did notice that there were some of the figures in chapter 2 of your new book that is not there in the last book that I had. I've not been supplied with the PowerPoints or anything of that nature, so I did not include them, but they are really good. But the concept will come again. So if you're going to ask me, hey, I'm going to spend this 20 lectures time or month and a half, read the new book. Don't waste your time for the old book. Okay? Because that's important for you. The old book, it was 2009, 2015, tons of things have changed. The concept would not change, but the things would change. I would never ever recommend you to read the old book. But I do see people want to go, and I think they are available rental, and the good thing is it's available even free online. So keep that in mind, okay? And I also noticed that some of the figures are kind of staggered. This follows from last year, from the old book. So figures are a little bit staggered. They have kind of replaced some figures with newer ones. But I will try to cover that up in the clinical part, as I have already told them to supply me with the PowerPoints. And I will help you. So the thing is for innate immunity, if a bacteria goes into your system, and this is unknown to the immune system, right? So adaptive immune response would not do anything, but would you let the bacteria or microorganisms stay in your system? No. So there will going to be a fight by innate immune system. So these are the two concepts you have to have, that endocytosis and phagocytosis, which are normal processes of cells to survive. They would eat food. They have to have, you remember we talked about electron transport chain, mitochondria, and biochemistry. Energy, ATP, is to be produced, but the two important things that you need is glucose and you need oxygen. So there's a respiration taking place at that level. Cells need it. But the same mechanism is used for example, say endocytosis. Many a time, the important concept over here, the two important concepts, one is pinocytosis for bigger molecules, macromolecules. And then there is another concept which is receptor-mediated endocytosis, and that you pretty much know when we talked of neuromuscular junction. We talked of different hormones, even insulin. Things don't just go into the cell. There has to be a receptor there. So that's again a very important concept of immunology. That all doors are locked, right? And all need a key. There's no keyless door. So you have to keep that in mind in terms of receptor mediated. But you can see this slide tells you there's a receptor, doesn't say which receptor, and there is a macromolecules. That's another way of bringing things in. And if you remember pathophysiology, if you do not, you can go back and read it again if you are interested. I have recorded all those lectures from physio. You can ask me for recorded lectures as well. I will also try to post it on this YouTube channel. But the concept I told you at that time was anything that comes in has to come in a vacuole. So there's a vacuole over here that is formed. That vacuole is called endosome. So that's like an endosome because things are not left loose. It's kind of covered. And also remember I told you that there are lysosomes that carry killing material. And I also told you the biggest concept of endosome-lysosome fusion for digestion to take place, okay? And that's what's happening over here. Now in this case, also keep in mind that let's say the same mechanism goes after a bacteria. So bacteria goes over here. So the endosome in this case will be called phagosome, pretty much the same thing. So it's going to have bacteria. We don't let bacteria to sit in the cell. So it's very much contained, covered. And then there are lysosomes which are filled with lytic enzyme. They fuse. So these lytic enzymes are released into and kill the bacteria. So this is a non-specific immune response that happens at all times. And if you remember when I taught you necrosis and apoptosis, I asked you this question: what is pus? What is pus? Dead white blood cells. You're right. Dead neutrophils. So if this fight is going on between neutrophils and the bacteria, in the process, there's no win-win situation. Bacteria are smart enough, you'll see it in the bacteriology. They kill neutrophils, and neutrophils kill them. So in the process, you end up with a pus. Okay? So also keep in mind that there are millions of bacteria, and so do the number of neutrophils. Okay, so but this kind of a mechanism that we have is important. And I also told you that when I talk of pathogenesis, something going wrong, I'm going to talk about things that can go wrong. I mean, you can just kind of, you can buy a new car, everything is perfect. No, the brakes show some problem, engine show some problem. There are many things that can come up. So right now everything is okay. But let's say if this receptor is defective, things are not okay. Let's say if these vacuoles within the lysosomes are not there. Let's say this fusion in lysosome does not occur. Some of the bacteria is pretty smart. They will go into the system, they will release something, and they will fool this lysosome. This lysosome would never come in contact with this phagosome, suggesting that this bacteria is immune, it's not going to get killed. Okay? And I don't know whether you remember when I taught you this, there was one bacteria I said, keep in mind that is one of the notorious bacteria that has a mechanism that would not allow phagosome-lysosome fusion, and we are still struggling for that. Can somebody name that bacteria? HPV is a virus. E. coli, no. You can pretty much fight E. coli. That was my question when I said Mycobacterium tuberculosis. Mycobacterium tuberculosis, because that's still a big, big problem in the world. Not the only mechanism, but this is one of the mechanisms that these bacteria have to resist that. So what it will mean is that you pick up a mycobacterium in Africa or Asia or any part of the world, and it sits in your cell somewhere, right? And then doesn't give you a disease. 20 years later, 30 years later, it gives you a disease. It is in the system. You didn't acquire it here. You brought it. We call imported diseases. Okay. So these are important concepts that I want you to understand. I'm pretty firm that whatever we discuss in these lectures, and you go back and try to at least read that chapter once, so it will kind of make sense to you. Okay. Now, as I said the other day, there are quite a few cells involved. Immune system, I just talked about polymorphonuclear neutrophils. We talked about cells. But remember, we are all cells, systems, tissues. By the end of the day, everything starts from one cell. That's what's called stem cell. Everything starts from a single cell and then divides into cells. And there are cells sitting in different parts of our body, and they have a name, and they are there for a good purpose. For example, macrophages, we talked of inflammation, acute and chronic inflammation, M1, M2. And then there are Kupffer cells. They are in the liver. They are alveolar cells. Of course, they are in the lungs. There are splenic cells. They are in the spleen. There are peritoneal cells. They are in peritoneum. There are glial cells. They are in the brain. So there are different cells in different parts of the body. But once they become permanent resident of those places, we give them a name. So they pretty much stick there. But then there are other cells that patrol. There are other cells that move around. I gave an example for neutrophils. I gave an example from red cells. And we're going to talk about B and T cells. Dendritic cells. So these are those kind of cells with dendrites, like a starfish. And these bear a different name at different places. When they are in the skin, they're called Langerhans cells. And I think when I taught you skin, I told you that we normally test your immune response, your allergic response, by applying antigens to your skin. We can do for mucosa, but mucosa is dangerous. If I was to test your allergy reaction by exposing antigen to mucosa, I have to do it in a clinic because things can go out of control, right? So asthma testing cannot be done at home. So it has to be done when you're prepared for a very severe hypersensitivity or allergic reaction. So skin is there. And this is the cell which actually is, if you remember pathophysiology, it was called professional antigen presenting cell, APC, right? Then there are natural killer cells. I'm going to discuss that in detail, as the name suggests. And they are natural killer T cells, suggesting that there could be natural killer cells which are not T cells. That's what it means, okay? Now, this is a slide of a blood slide. You can see a neutrophil surrounded by erythrocyte, just a peripheral blood film. And what you see is that, of course, what's the difference between a red cell and neutrophil? What is the difference between the neutrophil and a red cell? Sorry? Correct. Red cells do not have a nucleus, whereas these cells have a nucleus. And when I say nucleus, this pretty much mean that you have this all, transcription and signaling pathway outside in and inside out. But you can see this is a polymorphonuclear because the nucleus comes in different form. This one is a polymorphonuclear. Bilobe, trilobe is a different kind of nucleus over here. This is how a typical macrophage looks like. And what you see over here, the ruffles are spikes. Basically, these are receptors. These are very specific receptors to such an extent that, like in human body, their eyes, nose, ears, fingers, blah, blah, all different parts of the body. So these are very, very specific receptors, and we know most of them. And they are identified based upon these receptors, and they look like microvilli in this case, but this is an electron micrograph. Now, if I was to make a cartoon of that, now remember, again, the concept is, let me put it this way. If you remember me saying that each and every part of your cell body has an ID, right? So all different part, liver, spleen, you name it, has a tag, has a computer property tag over there, which is a bar. And then there are specialized cells which have a scanner. They're going to go scan it. Okay. Now, I also told you this kind of surveillance is taking place at all times. So for this, most of the time you will see that, if you see even in the grocery store, you have to bring scanner closer to that, right? You can do it from distance, but you have to close to that. So these cells have to come close to that and interact, and then make a decision what's going on. But that's very much regulated, very much controlled. And also keep in mind, I gave you the difference between self and non-self. So these cells are important. I'll discuss that in a subsequent lecture. There are sets of cells in our body, which are T cells, and they are educated in intrauterine life, and they are given those IDs of each and every cell. So they have a memory, and they can identify each and every cell. And they keep it with them for good for the rest of your life. So this is called education. They are educated for that. Okay, and they are the one who decide. And this all is there within your body. There is no external maneuvering. So that's why the concept of immunology is that you have to understand yourself, your own defense system, before you want to come and help the immune system. You don't want to give a drug or vaccine, anything. You don't know what's happening there. Maybe the whole overarching hypothesis is that we are all already well equipped to fight cancer, infectious disease, so on and so forth. To such an extent that whatever drugs we are trying to give it to you, your body already produces it. If we're going to help you out with treating depression, for example, we're going to mess up your dopamine level. So where is dopamine produced? It's produced in you. Is that the point? So likewise, we are struggling to find the exact link, exact concentration. That's where the struggle is. So in this case, you can see that there is a killer cell. So you can see this killer cell has all this killing abilities, carrying all this weaponry over here. But things are not random. If it becomes random, then you start killing yourself. There's a problem there. And that's the problem with autoimmunity. Your own cells are killing you. That's why there's no treatment there. What can you do? You cannot separate yourself from yourself. Does that make sense? So in this case, we are studying them. Let's say, for example, on the top, there's a killer cell. It has two kinds of receptor. It has an inhibitory receptor, right? So it's given a break. It will come and talk. We call this talking. Cells talk to each other. They come and talk to each other. So there is a cell, and this is a normal cell. This is your cell. So this will come close to it, and this cell will ask the normal cell, "Hey, who are you?" So they say, I'm you. I'm cell. So show me your ID. So he pops out this piece of paper and shows SS number. Okay, whatever. So it shows a ID which is very specific ID, should not be given to anybody else. That's what I say, SS number. And this is gene. We call major histocompatibility gene. That's very unique. That's why what it shows is that it has a cell and shows MSC class 1. So this shows a histocompatibility gene, which is 1. I'll discuss the full chapter on that, lecture on that. And then the killer cell will look at it and they say, "Hey, yeah, you're right." He said, "I'm going to take it." Say, "I'm not going to kill you. No lysis." Okay, you're fair and scared. Now, on the other hand, there is another cell, which is you, but unfortunately, this cell get infected with a virus, or this cell is a cancer cell in the same system. So the other cell will come over here. So it's going to look for ID. Now, remember I said as a basic concept, unfortunately, if there is no ID provided, right? So there's going to be no inhibition in killing. You say, oh, you don't have an ID, kill it. Because they are natural killer cells, so they are equipped with that. So they are going to kill it. Cell is dead. Okay, but also keep in mind, who told us that? We studied cancer cell and say how the cancer kills you, how the virus kills you. So the viruses of cancer are teaching us. In this case, what happens is that if, for example, a virus wants to survive or cancer wants to survive, it's going to take our own camouflage, take our tissue. It's like stolen ID. If okay, hey, this is me. It's going to provide that. There's nothing you can do. The killer cells will take it. It's not going to verify that. And we have a problem. Okay, so it's like more of an identification. So this step means that there are particular cells, but again, we talk about innate immunity. Does that make sense? Yes? Okay. Now, let's go back and forth for inflammation. We already discussed that. We talked about acute and chronic. That again is a part of innate immune response. I don't want to go in detail, but one of the things when we talked about inflammation, we talked about the cytokines, kinins. We talked about tons. I told you, whole pharmacy is based upon inflammation. Everything ends on inflammation. That's why I said that anything that ends with itis is inflammation. Infection, inflammation. Cancer, more or less inflammation. And then again, the only thing we want to make difference is whether it's an acute or chronic. That's where our dealing will be. Now, if you look at acute response and tie it with inflammation and tie it with innate immune system, you will again see fever. And then these are some of the things which are released in fever, in acute inflammation. So the initial infection that you get, and that's why the physician says, hey, you get a sore throat. Is it fever or not? How high is the fever? So he's looking whether your immune system is kind of going towards inflammation. It's going to put up a fight. Right? So that's good for you. So fever is good for you because what it means is you have a patent immune system, there's a response. Had there been no fever, then there's a problem there. So all those things, those look bad: pain, redness, swelling, fever, but if you look deeply, they're good for you because your system is putting up a fight. But then again, things can go out of control. Now, as I said in the previous slide, receptors, so the cells have to present receptors, and I told you, if I was to tell you more than 1 million receptor on one cell, that's the most difficult problem, difficult thing that we are having in cell biology. We are coming up, it's so complicated, right? And then remember, it's just like a jungle there. So we are trying to figure out which one actually is the one that we need to target for development of drug or development of many different things. So that happens as an important aspect. I'm just give you some examples. As I said, pattern recognition, receptor, I give an example of this computer ID, which are bars. So there's a pattern over there, but just by looking at it, you cannot differentiate. All the patterns would look alike unless you have a scanner there. But body has a system. Okay, so they are toll-like receptors which are expressed on extracellular pathogens, and then there are nucleotide binding domains we call NOD. They are also receptors. Then there are mannan binding proteins. They are also receptor. Then there are scavenger proteins. They are also receptor, and so forth. So what I'm trying to tell you is that there are very specific receptors. Some are more important than the others. Okay, and we need to engage those receptors. Right? It's just like I'm gonna extend my hand. So I'm expecting that he's gonna extend his hand, and that's what we're gonna shake a hand. If I extend my hand, he's not gonna bring cheek to me, and there's always a corresponding thing. So we want to find out where is the beginning of that handshake. Okay? So these are receptors expressed on the cells, and they are involved in innate immune response. What does that suggest to you? It suggests to you that if these receptors are expressed, you're going to go for a kill regardless of what the organism is. As long as they're expressed, let's go for it. Random. So there is a mechanism, that's an innate mechanism. The reason is because we are exposed to tons of microorganisms at all times. This pen, the tip of this pen, if I was to ask you to just put it on an agar plate, you'll see it's going to grow millions of bacteria. While you guys are sitting, all of you carry a commensal bacteria, which is your bacteria at all times. And you basically are sharing this space over here, environment, so you basically are sharing that at all times. That's why I say, hey, you are sick, stay home, because you want to share your virus with the next person. So we are already exposed. There's nothing you can do about that. The only thing you need to do about that is you have to have a normal immune system. That make sense? Okay. So when I say receptors, of course, these receptors have, and they actually look like the computer. It's funny thing is they look like this, all these computer bars. And they have a very specific kind of structure. And just like serrations, just like keys, they all look alike, but these are the difference over here. So I have all this departmental key, they're all the same, but I cannot tell on this number there, but these are serrations that will make the difference. So exactly these are like biochemical molecules. They all have attached to cell membrane, so they have an intracellular component, they have a transmembrane component, you can see, and there is something jutting out, extending out in terms of layers, okay? And they are important. Some of them are expressed on your cell membrane, and some of them are expressed on the bacteria. So this is how the whole sequence happens. They need to talk to each other. Now, if you pay attention to the top, over here, say gram-negative bacteria. So bacteria that are gram-negative, they express, those receptors which are LPS receptors. Then if they are gram-positive bacteria, example is mycobacteria. I just give example Mycobacterium tuberculosis, right? So they express these kind of molecules over here. These are lipopeptides, liponucleic acid, lipoarabinomannan, and they're very, very important. Why they're important? Because they're expressed in the cell membrane, and basically all the drugs that you're targeting in terms of antibiotics are going to go for the membrane. Okay? And if it happens on a viral and bacteria, so they have nucleic acids. So they have very double-stranded RNA, single-stranded RNA, so on and so forth. So you can see that things don't happen randomly. What I'm trying to say is that your system has a very precise mechanism for looking at, hey, I'm going to fight with bacteria. Which kind of bacteria? I'm going to fight with viruses. Which kind of virus? I'm going to fight with fungi. Which kind of? Very specific. Very, very specific. It's good, though we can have a very targeted therapy. We cannot give like a journal antibiotic, let's kill everything. You cannot do that. You have to be very specific. And the example is that you can see this particular bacteria, it comes with those kind of receptors, right? And then some of these receptors, which are microbial polysaccharides, like sugars, they are basically seen by some of the other receptors, which we call mannose binding lectin, and they basically talk to each other and bring them to justice, so to speak, or bring them to the cells which will take care of them, right? So, because the whole idea is that system has to be patent. If the system is not patent, then you're going to take your own cells and kill them. Okay, so we are putting up a fight. That's the whole concept from microbiology, immunology. So I'm going to teach you immunology, what is normal, what is there in you. And when we talk of microbiology, bacteria, say what the bacteria has to offer. and then we'll have confrontation. We are learning from them, we are fighting them off. But they are very smart, very clever, and you can see things are changing every day. Okay. Now, cells and organs of acquired immunity or adaptive immunity, I told you lymphatic system is there. When we say lymphatic system, we usually talk of two types of organs. One is primary lymphatic organs, one are secondary lymphatic organs, meaning that secondary may have a second job as well. Primary have only one job, and that's lymphatic system. So the primary one is thymus. And the secondary are tons of them. There are tons of them. Secondary, you can see spleen. You have lymph nodes spread all over your body. That includes tonsils, Peyer's patches, mucosa-associated lymph tissues. Then you have gut-associated lymph tissues. Then you have other membranes, or you have other cavities in your body, in your liver, in your brain, right? In your meninges, they are associated. So they are all association because everything has to be reconciled with the immune system to stay in place. Okay. Now, anatomy, you can see from here, primary lymphoid organs, and most of the time we consider that as a thymus, which is somewhere over here. Over here you can see. And bone marrow is also considered as a primary lymphoid organ because that's where these two B and T cells are developed. T cells mostly from thymus, that's what called T cell. B cell basically are from Bursa fabricius, that's in birds. That's what we call B cells. But for you, even if you say bone marrow, that's fine with me. But historically, they are from Bursa of fabricius that was discovered in birds. Then I said that all the body parts are supplied with lymphatics, and all these lymphatics and blood, they talk to each other. And then it's pumped into right subclavian artery. We talked about thoracic duct, typical that's circulation. So lymphatics and blood, they kind of run around. There's a special circulation where lymph is running, special circulation where blood is running, lymph drains into the blood, and so on and so forth, and the whole system goes on. If you look at thymus, for example, so if it's a section of a thymus, you will see over here that there are cells there. So you will see that there are special cells for thymus, which is called Hassel corpuscle in the medulla of the thymus, and then there are some other cells which are there, and they have a special purpose. This is where the cells have been produced. In most of us, thymus gets involuted. This means dries out at birth because T cells have been made for good done. Okay, but sometimes it stays. There's a problem there. Spleen, if you can, spleen that purifies the blood. So blood is pumped through the spleen at all time, and this spleen, you can see arterial blood and venous blood. You can look at in the cardiovascular system, and you will see over here that cut section of a typical lymph node where it has a cortex and medulla, and that's where you will see that there are B and T cells. So you can see interfollicular T cells, and you can also see B cells. So all these B and T cells are those lymph nodes. So once you get infection over here, your lymph nodes get swollen. This means that more supply of B and T cells to produce them, they get swollen up. So these are the lymph nodes where those cells are produced, and different parts of the lymph nodes. So you can see these are, and the same cells over here will have some special cells like antigen presenting cells. So what will happen is the T cell will pick up an antigen from periphery, bring it to the tonsils, right, and present it to an antigen presenting cells. And antigen presenting is going to run a background check whether you've been exposed to it or not. If you've been exposed to it, the information is already there. Say, hey, we've already seen this 10 years ago. We already prepared antibodies for that. Let's bring them in to fight that. So this is an adaptive immune response. That's the concept of vaccination that we normally have. Now, the question is, when I say antigen, so all those molecules expressed on your cells are also antigens technically, but your body knows them. So this is you. So they're not going to get killed. But anything foreign outside, which is coming to you, whether you eat, drink, touch, whatever way it touches you, is an antigen for all practical purposes. Now let's see, for example, what routes do we have? We have a physical barrier, skin and mucous membrane, right? So you get a prick. You get a prick, and that antigen, let's say it's a hepatitis virus antigen, let's say, is going to go into your blood. From the blood, it's going to go into circulation. It's going to go into spleen, and spleen will look at, the T cells will look at whether you have antibodies. If you've been vaccinated for that, you already carry antibodies for that. So antibodies will fight with this antigen, right? But this is one way, the other way is that you basically get antigen from the skin. So the antigen comes in the tissues, different part of the skin. So all the skins is being surveyed by immune system. There are cells over there, they will pick up the antigen and want to react or do not want to react. Just like tuberculin test. That's why if you have a BCG vaccination, United States is the only country in the world that doesn't kind of force BCG vaccination, the rest of the world does. So at birth, everybody is given a vaccine which takes an antigen of MTB to teach your system, hey, this is how the MTB looks like, be careful, prepare something for it. Not in US, okay? The third thing is that the antigens basically can go directly through the tonsils. So you have these tonsils over here. And then again, they are kind of being circulated. So lymphatics, they all drain into thoracic duct. Thoracic duct goes into vena cava on the right-hand side, and then it goes to the blood. So there is a continuous distribution of lymphatic system and blood system at all times. There's nothing that can escape at being identified and being kind of processed, okay? So what happens is, as I just said, that fate of antigen is, goes in the bloodstream, it follows the track, it goes to the skin, it kind of circulates. If it goes in mucosa, pretty much the same. All these things will allow the antigen to come across. It's just like you want to enter United States by air, by sea. There always will be ports, and there's going to be a checkpoint over there. You want to go through a process of immigration. They're going to ask you a document. That's exactly the case in the body, and very, very specialized cells over there. They keep all the information. They have all this shared information to make sure that you do what you need to do. Okay. The other term I'm going to kind of introduce today is that lymphocyte. When I say lymphocyte, I mean both B and T cells. Now, the term I'm going to use is naïve. Is it French or German? Is it French? Okay, I'll take your word for that. Okay, so this means, hey, you're too naive. You silly, foolish, whatever. You don't know anything. So there are cells. The other term, immunologic terms, are virgin. They are those cells, they have never seen an antigen. So they don't know. So they are naive cells. They are there because non-immunized, millions of them. But they have the ability to recognize. Then the thing is this, that nature has designed a mechanism that you encounter an antigen, and within 72 hours, you're going to get a response. That's why when we give you a tuberculin test, right, you need to respond in 3 days. That's what 72 hours is coming from. Because it's going to get circulated in a T cell, system say, hey, yes, I've seen that antigen. When I was a kid, I have a BCG. Oh, why did you not tell me that you were BCG vaccinated? Because all those people who are BCG vaccinated should tell the physician that they should not give them tuberculin test. Because tuberculin test is for those people who have never been exposed to TB. Okay, so within 72 hours, if you do that, you will have a very kind of tissue destruction and reaction there, because your immune system is going to react to that. Okay, now the bottom line is that I want you to think in terms of the way we explain to you. I think some of you may feel, hey, either it's going to be innate or acquired. Yes, true, but again, there is a overlap there because the players are the same. The players are the same. So you can see from innate on the top to acquired or adaptive, they're basically all there to help you. And in innate, we have physical barrier like skin and mucous membrane. We have tons of cells which are non-specific. We have chemical barriers, I told you that, right? Whereas in acquired basically has two hands. One is humoral antibody, the other is cells, right? When I say cell again has two hands. One is a B cell and another is a T cell. So you can see from here that there are B and T cells, but as I said, that they are very specific. They are very specific. So what happens is, even, I will give an example of like this homeland security mechanism. So if somebody was to, even if you go for immigration and those people over there, they bear a tag, identification tag themselves before they're gonna ask yours. So it's just like somebody knocks on the door, hey, I want you to give me your card, this, and so he has to show his identity first before he's going to ask you. This is your right. Exactly the same in the body. So each and every tag system, but this tag system is not given to these cells per se, specifically. There was a time, I remember 10 years ago, we used to have you register according to your SS number, and we used to have SS number, but then realized it's not good. So now we have your student ID number. But still, there is somebody who keeps your SS number. Does that make sense? So exactly the same way, very specific, nothing can miss, is this antigen-specific receptor, which is B and T cells. Okay? And the way they put up a fight basically is called immune response. And that is the basis of disease. Any disease is foreign. Any disease, you should not have a disease. That's the concept. You can say, hey, I'm normal, but I'm sick. That's not possible, right? Well, having said that, some people know that they have chronic inflammation, allergies, and so on and so forth, but still it's not good, okay? So we need to understand normal before we understand abnormal, okay? So I'll stop here.